online event of the Affiliation of Christian Biologists and the American Scientific Affiliation. My name is Brian Greuel, and I am the president of the ACB. Tonight, we are delighted to have Dr. Francis Collins as our guest speaker. Dr. Collins is a physician and geneticist known for his landmark discoveries of disease genes, for spearheading the Human Genome Project, and for his 12 years of leadership of the National Institutes of Health under 3 U.S. presidents, Obama, Trump, and Biden. As NIH director, he guided the nation's biomedical research in everything from basic science to clinical trials, including a dramatically successful series of research partnerships addressing the COVID-19 pandemic. After stepping down from that role in December 2021, Dr. Collins served as acting science advisor to President Biden, and later as a special advisor to the president for special projects. He continues to serve as the White House lead for a bold program to eliminate hepatitis C in the U.S. while leading his research group as a distinguished investigator in the National Human Genome Research Institute. Dr. Collins is also very well known for his unwavering promotion of the harmony between science and faith. In his book, The Language of God, A Scientist Presents Evidence for Belief, he describes this personal spiritual journey and how he integrates science with a Christian worldview. He also assembled an anthology of selected readings on faith and reason entitled Belief. Shortly after his first book came out, Dr. Collins founded the BioLogos Foundation, which is a well-respected faith science organization. And he served as its first president until he was appointed as NIH director. In 2020, he was named the 50th winner of the Templeton Prize, which celebrates scientific and spiritual curiosity. Perhaps most relevant to tonight, Dr. Collins is the author of the book, The Language of Life, DNA and the Revolution in Personalized Medicine. Tonight, he will speak to us about the promises and challenges of implementing precision health. Dr. Collins, thank you for joining us this evening.
Well, thank you, Brian, for a very kind introduction, and good evening to all of you, the 74 people that have signed up for this presentation. And I'm grateful to look across the names I see on the Zoom and recognize a fair number of people who I know and am honored to be friends with and colleagues with. I know the Affiliation of Christian Biologists is a subgroup of the American Scientific Affiliation, and I really appreciate all that you are doing. And the American Scientific Affiliation was really helpful to me when I first became a Christian at age 27 and was trying to understand how other people who were interested in science could also be followers of Jesus. And ASA emerged as a really good answer to that question. And I went to my very first gathering of scientists who are also Christians at an ASA meeting in Boston, must have been about 1978. I became a member and have been so ever since, and happy to be a fellow of the ASA as well. So I'm going to talk about science and medicine, but I will also not be shy about the fact that from my perspective, this is all deeply connected to something even more substantive, which is that we are creatures of God's creation. We are children of God. We are followers of Jesus, and all of this needs to fit together and does fit together in really remarkably harmonious ways. And I'll fold that in a little bit here and there into what I'm hoping to share with you. And I'm looking forward to the Q&A when we get there. And I know Brian will give you more instructions at that point about how to pose questions. But given my challenge here of talking about precision health, let's see if something useful can come up from that particular assignment. So yes, the topic, promises and challenges of implementing precision health. So what are we talking about when we're talking about bringing more precision to health? This is basically an emerging, not fully yet realized approach to disease prevention and treatment, just not about treating disease, but also preventing it, that intends to go beyond what has for most of human history, been a one-size-fits-all approach, at least in Western medicine. And replacing that with medical care that's designed to fit our unique medical selves, which doesn't just focus on genes, although I will talk about that a fair amount, but also on lifestyle, environment, health behaviors, and so on. And I think it's hard to argue with the idea that this is a good thing if we can do it. You wouldn't go to the drugstore and pick out some random set of glasses to try to help you. You'd rather have prescriptions that are suited for your eyes. You wouldn't submit yourself to a blood transfusion that hadn't been typed and crossed to match you. But a lot of what we've been doing otherwise in medicine has not taken advantage of those kinds of individual special approaches and have been pretty much generic, one size fits all. Let's see if we can do better than that across the board. But that needs lots of insights and technologies and science. And we're in the midst of seeing that kind of expansion of our understanding start to happen. So I'll be talking a lot about DNA, but I also want to say from the get-go that for me, as somebody who is a geneticist, but also a follower of Jesus and someone who understands the nature of God's creation, that studying how we are all biologically put together is also an opportunity to see the hand of the Creator. And in that regard, the laboratory is also kind of a cathedral where we get the chance to understand how this creation in its incredible complexity and elegance is a reflection of the mind behind it. I'm very fond of this comparison of these two images. On the right, you see DNA, not in the form you usually see it, looking at it from the side, but looking down the long axis of the double helix and appreciating its beautiful symmetry, almost reminiscent, perhaps, of something like a stained glass window here represented by York Minster. So throughout this, as I'm talking to you about scientific advances, I hope we can also reflect a bit on what that says about the incredible gift we've been given of a creation that is elegant and beautiful and truly complex. So here's what you're in for in my part of this, which will go on probably for a bit until maybe about 8:30 Eastern, and then we'll have time for questions from you. I do need to talk about the Human Genome Project because it's sort of how this whole revolution, if you want to call it that, in genomics and in omics, because it's lots of things besides sequencing DNA that now find their way into this, got underway. We'll then talk about how this applies to common diseases and to the whole topic of GWAS, genome-wide association studies that are shining a light into hereditary factors in really complicated human conditions that previously were not very within our reach. Then I'll talk about the clinical applications of all this. I will be doing an advertisement for a program called All of Us, which is the most historically challenging and potentially significant large-scale cohort study ever mounted by the National Institutes of Health, and perhaps encouraging you to think about joining, which I, as a member myself, I think you would find to be an interesting thing, but you can decide. And then I want to go not just from predictions in basic science, but into therapeutics. That will raise some ethical considerations that we need to chew on. And then finally, all of this is great in terms of the medical research, the advances that happen from clinical trials, but how do we actually get the cures to the patients when we know something works? We're not altogether there yet either. I'll have something to say about that. So that's what you're in for. And I will try to walk us through this step by step so you don't get lost. And again, try to stick to the time so we have plenty of opportunities for questions. So the Human Genome Project. Those of you who've been around a while probably recall in the late 1980s when the idea of sequencing all 3 billion letters of the human DNA instruction book was being discussed. It was not all that popular. There are a lot of people who said, this is a dumb idea. This is going to be way too expensive, probably technically unfeasible. Maybe it will just, recruit really people who aren't that smart anyway, because it sounds sort of mind-numbing to just keep reading out base pairs from the DNA sequence. But it was hard to argue that you wouldn't want to know that information if it was possible to obtain it. And so the Genome Project did, in fact, get underway. And in 1990, started in the US and in other countries, although the US had the lead both in terms of direction of the project and the resources put into it. I could spend a long time going through what happened over the next 13 years, and I won't. You can see in this diagram a whole lot of milestones that happened along the way, and that was really important. It was a project that was really carefully designed to have intermediate goals where you could decide whether this was actually working. And the actual sequencing of human DNA didn't really get underway until about 1997. And most of the sequencing of the human genome happened in the last 18 months when the technology was really ready for it. At that point, we were sequencing 1,000 base pairs every second, 7 days a week, 24 hours a day, which is breathtaking when you consider when we started out, you would be doing well to sequence 1,000 base pairs in a week. But thanks to the incredible efforts of a remarkably dedicated group of scientists, 2,500 of them, in 6 countries, working together in 20 institutions over 13 years, this ultimately did succeed 2 years early and happily under the budget that had originally been estimated for its completion. And I can't say enough about the remarkable group of scientists who did this work, not worrying too much about who is going to get the credit, just excited that they could be part of something historic. You see a picture of a bunch of us gathered there near the end of the project in a meeting at Cold Spring Harbor in New York. And in 2000, the summer of 2000, the picture in the upper right, you can see an announcement of the fact that the draft, at least, of the human genome was in fact in hand in an episode that happened in the East Room of the White House with President Clinton. But that was a draft. The description of that draft appeared a few months later in Nature magazine, and the completion of the human genome at that point in 2003, with all the technology that was available at that time, carried us to the point of having a very good sequence representation of a reference individual, actually a sort of a collection of reference individuals of anonymous sort. Interestingly, however, some cynics would say we keep reclaiming the successful completion of the human genome because there were some of those naughty bits there that were really hard to sequence. And I'm happy to say that I think we finally reached the point of saying, yes, it's really done. But if you look at the science cover here on the right, that's 2022. That's just 2 years ago, where with new sequencing technologies that allow you to read across very repetitive DNA, sometimes over tens of kilobases, you can actually get the whole thing done. And that's the only way we could really get across the centromeres and some other repetitive sequences. So the gaps have now been filled, and we have a complete, as they say, telomere to telomere human genome. In fact, many such genomes are now being produced in a pan-genome project that's going to teach us about human variation in exquisitely detailed ways. But of course, that's a sequence written in a language that we don't understand all that well. Some people thought, well, once we have the human genome, everything will change overnight. They weren't really paying attention to the biological challenges here, or to the fact that less than 2% of the human genome actually codes for protein. The rest of it was perhaps in an unfortunate way called junk, and it certainly is not. The rest of it is where all the regulatory information exists that makes it possible for this same instruction book to direct the behavior of a liver cell or a brain cell or a bone marrow cell, all of which have that same book, but need to use it in very different ways. And a lot of the effort right now is focused on trying to understand how that regulatory part works. So we had the sequence. For me as a physician, that was a milestone, but not nearly good enough because you wanted to take this to the point where it was actually going to help somebody and where you would have at the bedside the opportunity to examine things like a genome sequence and other kinds of omics data in a way that would be useful, not in the way depicted in this cartoon where everybody's like, I don't know, what do you think? So a lot of work had to get done. So immediately following the completion of the genome sequencing effort, a whole lot of other projects were developed and supported, especially by NIH, but also by other partners, to try to learn more about how the genome actually works. And I'm not going into each one of these many different projects, but they were all multi-collaborator efforts where they agreed to give their data away as soon as they had it. That was a ethic that emerged from the Human Genome Project. If you're doing a large-scale collaborative effort on our shared inheritance, then you got to give the data away. This is not the kind of thing where you hang on to it and try to make some sort of personal benefit from it. So all of these were of that sort, and that whole ethic has really been sustained in the field of genomics in a wonderful way. In addition to all of these projects, which were focused on trying to determine the functions of the genome, there was also a lot of effort to try to use the information about the genome to discover the causes of disorders that we knew were inherited in a fashion that Gregor Mendel would appreciate. They were recessives or dominants or X-linked, but most of them we didn't know the actual gene involved. All of that changed. Look at the way this curve just rocketed upward, starting in the 1990s. And then once the genome was really completed, moving forward very rapidly, we now know the molecular basis at the DNA level of close to 7,000 disorders that are inherited in a Mendelian fashion. Most of those are pretty rare. But if it's your family, you don't particularly care if it's rare. You want somebody to work on it. And at least this gave us the chance to figure out how we could identify the causative gene, and that would provide a diagnostic test to figure out how to best identify who had the condition. So that was sort of the good news. The not so good news is that of those 7,000 diseases, even today, perhaps 500 of them have some kind of approved therapy. And oftentimes those therapies are far from what you would hope for. They are imperfect. They may extend benefits a bit, reducing symptoms or extending life, but they are, for the most part, not cures. So we have this really big gap between diagnostic abilities and therapeutic abilities. And I'll come back to that in a bit because I want to talk about where we are with having the genome become also the source for ways that we could actually cure diseases and not just diagnose them. But first, I want to talk about those disorders, which are even much more common, which are not Mendelian, but do have hereditary contributions, what we traditionally call common diseases, or somebody would call them polygenic diseases or complex diseases. I'm talking about diabetes and heart disease and the common cancers and the common mental illnesses. We know those all tend to run in families, but not in a way that's easy to understand on the basis of single genes being involved. And before we really knew, I think a lot of people hoped that maybe those would also be relatively straightforward. Take diabetes, which is what my lab primarily works on, type 2 diabetes, the adult type. A lot of people speculated that there might only be a half dozen genes that would be involved in the risk, but even a half dozen is a lot more than one. And we didn't have the power in our methods to be able to uncover this over the course of the preceding years. And then GWAS came along. So I need to talk about GWAS, genome-wide association studies. So this is an approach which is actually very powerful, but could not be done until we had a really good characterization, not just of a single reference genome, but of lots of genomes, where we could start to understand the differences between individuals, those variations that happen about 1 out of every 1,000 base pairs. You might have a T, I might have a C in that space. Somebody else might have down the line an A where I have a G. Most of those are probably bland variants that don't make any difference, but some of them fall in a place that plays a role in the risk of a disease. Most of them in a regulatory region, not necessarily in the protein coding part of the genome, but in that regulatory part, which is most of it. How would you find those? Well, basically, you'd want to come up with a strategy that allowed you in a case control fashion to identify that a particular variant appears a little bit more commonly in the people with the disease than the people who don't have it. This slightly blurry example is supposed to show you how that works. You have controls, you have cases. You basically look across the whole genome, and the methods to do that have gotten increasingly cheap and rapid. And you look to see with a SNP array, a SNP being a single nucleotide polymorphism. That's a single letter in the genome that might be a T or a C, depending on who you're measuring. You look to see, is there a difference? And if you do it right, and your cases and controls are well matched so that you're not picking up something else which will confound your analyses, and there are ways to be sure about that matching, then you're going to find across the whole genome, as you see down here in this so-called Manhattan plot, places where there is a difference between the cases and the controls. And here we've got this mapped across the chromosomes 1 through 22. And in this toy example, there is something going on in chromosome 1 here where there's an association that seems to, in the limit of this experiment, to reach statistical significance. Now, I will say this is an old slide, and the limit of statistical significance gets steeper as we get more rigorous about this. But that's the idea. It's a case control study. You're looking across the whole genome. You're trying to say, are there places where there is a variant in the case group that is slightly more common than in the control group? And if you convince yourself that you have found that, then you've got a window into what might be an important pathway involved in that disease. Well, this was an idea in sort of the turn of the century, and then we had to develop the technologies to be able to actually do this. Again, my lab, working on diabetes since 1993, thought this would be a good thing. And here we were, the first indication this could work. This is now some time ago, 2007, 17 years ago, 3 papers in Science magazine. The third one here, I'm happy to say we were part of, basically looking to see whether this worked across the genome for type 2 diabetes. And 3 different groups coming up with answers that happily overlapped with different populations, which made you really believe the result was right. At this point, it was multiple susceptibility variants, as we said in the title of our article. Well, it was about 12. And we were pretty excited about that at that point. Well, that was then. Of course, if you really want to boost the power of this kind of analysis, then you want lots and lots more cases and lots more controls. So let me leap you forward to just last month. This is from the journal Nature from February 19th. A report that my lab was also part of. Now look, we've got 428,000 cases and over 2 million controls. Scanning across the whole genome, the same Manhattan plot. Look at the y-axis here, though. This is the negative log 10 p-value. So for instance, take the hottest signals here, which are up at the top. That's p-value of 10 to the minus 300, actually considerably higher than that. So this is real. You can be sure about this. And the usual way in which we decide this can't be a fluke is to have the p-value less than 5 times 10 to the minus 8, because you're doing multiple comparisons here across the whole genome. So you're not going to be happy with 0.05, but you could be happy with 5 times 10 to the minus 8. And the result across the genome, remember we had 12 in 2007, we now have 611 loci, that is segments of the human genome that contain variants that play a role in the risk of type 2 diabetes. This is maybe an overabundance of success, but it is what it is. We didn't know what the architecture of the genome was going to be, and this is how we find it out. And every one of those variants somehow is contributing to a risk of diabetes, most of them on a very, very modest level. But collectively, this seems to explain most of the hereditary contributions to this disease. I guess there probably, if we went to 10 million controls and 10 million cases, we might still find a few more, but this is getting pretty close. And those, of course, now are of intense interest, trying to understand how does that work. And that's a big part of what labs like mine are now trying to do. how to use that information to understand the functional basis of diabetes and how you could better prevent it and how you could better treat it, and also how you could take this disease we call type 2 diabetes, which is just this big lumped together description, and understand the subsets that are actually functionally and metabolically rather different, with different genetic risk factors, and in a way that would suggest better ways to be more precise. We're back to precision medicine here about how to prevent and treat them. And there's a lot of work going on in that space as well. So this is diabetes, but this is just one of many examples. GWAS can be applied to virtually any condition where you're interested in uncovering hereditary contributions, and you have enough cases and controls to be able to have sufficient power. Here's an interesting one. Groups decided to figure out with COVID-19, is there a genetic contribution that explains how some people handle it pretty well and others end up very sick in the ICU or even dying? And here was a study done by several groups, not with a huge amount of data here, because it was a very strong signal they found here on chromosome 3. You can see again the y-axis showing this is like a p-value of 10 to the minus 14, so believe it. And when you blow up that part of chromosome 3 over here on the right to see what's there, these points simply tell you which one of the variants seems to be driving the signal. And because they're all closely linked in something called linkage disequilibrium, it's not possible to say precisely which variant is the causative one. It's probably one of those right at the top. But when you look to see what genes are in this space of chromosome 3, oh my goodness, it's a hotbed of genes involved in the immune response. Kind of makes sense. So something's going on there. Maybe it's in fact the people whose immune response is overactive. That means they end up getting severely ill because the immune system kind of makes it worse by overkill. Or maybe they're the under-responsive ones. That's all being worked out. Just as a little aside, this is also a very interesting finding, because this piece of DNA on chromosome 3 is also one where about 30 or 40% of people in European backgrounds have a region here that's derived from Neanderthals. And this is a very interesting additional side study about how genomics is teaching us about our own origins. And it's the Neanderthal segment that seems to confer the increased risk for COVID-19 for reasons that aren't entirely clear. Certainly for those of us who are interested in science and faith discussions, the idea that there is clear evidence in our genomes that there was some mixing of Neanderthal and Homo sapiens DNA is of interest and deserves some reflection. But in this case, medically, it also seems to play a role in the response to this recent pandemic. And I don't intend to talk about COVID-19 other than this tonight, since we're talking about precision medicine, but I'll be glad if people have questions in the Q&A. So again, GWAS, once you understand the approach, can be applied broadly to lots of conditions, and it has been. Lots of labs, lots of clinical groups bringing their capabilities to the table here. This overwhelming picture is just the first 12 chromosomes, 1 through 12 from the left to the right. Each one of those dots there is a GWAS signal that somebody has convinced the community is actually real, relating to some condition. I won't tell you what the color codes are because I can't quite remember, but this is autoimmune diseases, this is cardiovascular disease, this is neurological conditions and mental health conditions. Everything you can think of has had GWAS applied to it. And the proliferation of discoveries is just phenomenal when you consider in 2006, there would have been like nothing on this chart. So that's good because that gives us insight, but how does that insight actually help? Well, if this is this chromosome and those are those colored circles that made a discovery, what do you do with that? Some of it's just basic science, figuring out statistics, figuring out candidate genes that might be involved in a disease, or what's the mechanism that the disease comes about. But some of it's clinical over here on the right. It is, after all, pointing you towards potentially druggable pathways. If I have a variant that increased my risk of diabetes, and I know that that's because I make more of a particular protein than I should, then if you develop a drug that kind of blocks that protein, that might actually prevent my diabetes. That's the argument. And there are good examples where that argument has played out positively and others where it's been disappointing. But it's also an opportunity to use prospectively to try to predict who might be at high risk in a way that would assist them in their own prevention strategy. We're not very good at preventive medicine. I think most of the time we kind of are doing this one size fits all, but there are exceptions. I would bet most of you know what your cholesterol level is. And you know it because you know that that is a predictor of possible cardiovascular disease. And it's not too bad at that. But we don't have very many like that that have been very, very well validated. Here's where genomics is going to be changing things, although the actual implementation of this has not quite happened yet. So let's talk about these clinical applications. One of them is this idea of a polygenic risk score, a PRS. I already told you there's 611 places in the genome that put people at risk for diabetes. And at those 611 places, you might have in your DNA a variant that makes your risk a little higher, and then down the line, another one that makes it a little lower. What you really want to know is what's the overall effect? That's what the polygenic risk score does. And if you imagine this sort of pileup of people who've had those risk scores calculated, most people are going to be kind of somewhere in the middle. But out there on the tail are going to be people who, by whatever luck of the genetic draw from their parents, have inherited a high number of these risky versions of those 611 signals and really puts them up at high risk. And some of those people will be at the kind of high risk that's kind of equivalent to a cholesterol measure saying you are really somebody who's on the path towards diabetes. That would be useful to know if you were 30 years old and trying to think about your diet and exercise. Because here's a disease where we know that the avoidance of the disease depends heavily on things that depend on the environment, namely obesity, diet, and exercise. We haven't yet gotten to the point of implementing this in medical practice. Why not? Well, because most of us have not yet had our genome sequenced and had that information placed in our medical record. But if we did, and if that was available right now, this is feasible today. This is not some future possibility. We could all be in a circumstance of having this kind of information about diseases like diabetes, where knowing you're at high risk provides you with an opportunity to take actions in terms of your lifestyle, your diet, your exercise. Now, would people actually take actions? We could argue a bit about that in terms of behavioral health. We as humans are not necessarily so amenable to that, but some people certainly are. And I think they ought to have the chance to know where they stand. And this would be taking the current approach to prevention, which again is pretty much one size fits all with a few exceptions like your cholesterol, and moving that into a much more individualized precision medicine kind of approach. That's one application. Another is this whole idea of pharmacogenomics. Look at this diagram at the bottom. You got this bunch of patients who have a condition for which a prescription is appropriate. Maybe it's heart disease, maybe it's depression. All of them seem pretty much like they're in a similar place. They get the diagnosis, they get the prescription. It doesn't always turn out well. You want to be over here in the lower right corner where the drug wasn't toxic and it was beneficial. But even for drugs that we have lots of experience with, that's far from 100%. There are other people, the worst situation, and the drug's toxic and it didn't help them at all. There may be some where there's some toxicity, but they still get some benefit. And of course, over here, where the drug is not toxic, well, okay, at least didn't hurt them, but it didn't help them either. Couldn't we do a better job of figuring out who's in which group? Well, a lot of this is genetics, because drugs are metabolized through pathways that we are increasingly learning how to understand. And so you can, in fact, and if you look FDA would tell you for more than 100 drugs, we know this kind of information. And you'll even see in the fine print of those labels that ideally the prescriber ought to know the individual's genetic state before prescribing this drug because it may have an impact on whether it's going to help them or whether the dose is right. Sometimes that's the other aspect of it. Maybe you're a rapid metabolizer and it's the right drug for you, but it's so quickly metabolized that you never get an effective level. Or maybe you're one of those people where the particular drug target isn't really there in the same way. And so you could take a big dose of this and still not get much of an effect. We know how to do all this for a lot of drugs, but we're not applying it. Again, how come? It's not that the data isn't there. It's that most of us don't have that data in our record. When your doc wants to write a prescription, they don't want to send off a DNA test and wait 2 weeks for it to come back to decide whether to write that prescription. They want to do it right then. And right now, that's not really feasible. We need to get there. So I'm talking about GWAS as though it is a big window into insights about disease, and it is. But let's be clear, a window for whom? Most studies that have been done on the populations where most research goes on, which is people of European background. If you look at all the GWAS studies that have been done on all these diseases, you will see 78% European, Asian 10%, and then after that, it gets to be really small. And that's clearly not a representation of our country, much less the world. So we have something we need to do here in terms of increasing the attention to diversity in our genome studies so that we actually can derive insights that are applicable to everybody. So here comes the commercial. Our US project to address this is actually underway, and there's other projects around the world to address this. But let me tell you about All of Us. So All of Us, as it's named, is this bold, unprecedented, longitudinal effort to enroll no less than 1 million people, maybe more, living in the US to accelerate research and improve health. The idea being to convince these folks to be our partners. They're not like human subjects. I hate that term, by the way. They're partners. They're participants. They are sharing in this adventure here, and they're getting all the data back about themselves that they want. Basically collect every bit of information you can on these 1 million people. Their health behaviors, they answer a lot of questionnaires. Their genomes are all sequenced. They basically make their electronic health records available so you can see what their medical exposures have been, all anonymized. And then using that, we have a chance to figure out with 1 million people who are highly diverse, how does this all work? How do genes and environment and health behaviors play out in a highly diverse, large group to determine who gets sick and who's healthy? Already 750,000 have joined. If you want to follow up on this, joinallofus.org is what you need to know because we've still got 250,000 to go. And there's plenty of time, but not infinite time, to come and join this. So let me say a bit more about All of Us and how it works. And by the way, the leader of this, Josh Denny, who I recruited from Vanderbilt to run this effort, just a brilliant physician scientist who's also a real expert in computational biology, is the CEO of All of Us, and also a very devoted Christian. Where are we? 750,000 participants have enrolled. They have various steps that they have to go through, and some of them are still going through it. So 517,000 of them have all of the steps done that you need to be part of the program. They're scattered all over the country. That was part of the point. And very importantly, they're highly diverse. 53% are white, so just over half, but most of the rest are a very diverse category. If you look over on the right here, UBR meaning previously unrepresented in biomedical research. So these are people who often don't turn up in biomedical research groups. 47% of race or ethnic minorities, lots of them with healthcare access limitations. Look at income here. 23% of these people have annual income less than $25,000 a year. So we're really trying to sample across the socioeconomic status. And multiple other areas also that are traditionally underrepresented, adding up to about 80% of the participants. Very intentionally here, trying to capture How are these factors about genetics and environment and health behaviors going to turn out differently depending on which group you're studying? If you want to be part of this, basically, you decide you're going to make your electronic health records available with lots of privacy protections. You answer a lot of surveys. You undergo some physical measurements. They're pretty simple. You give blood samples and urine samples. You walk around with things like Fitbits that you agree to have that data available. And then all of this goes into a cloud-based resource that is highly protected and which allows researchers who agree not to try to identify individuals to see what they can learn from this. And you get information back. You get information about your genome, about how you compare in various surveys, about how you compare in electronic health records and claims, other updates, aggregate results, scientific findings. You're really part of this enterprise. And you also can be recontacted if somebody has a great research idea for something that might be relevant to you. They can reach out to you and you can say no, but you can also say, yeah, tell me more about that. The kinds of information you get back if you're part of this, you certainly get genetic ancestry divided into 7 regions with some subregions as well. The kind of thing you might get from 23 and me or ancestry.com, except here it doesn't cost you anything. If you're part of this, this is all part of what you get back. A little bit of silliness about various traits. Is your earwax, is it wet or dry? I think a Q-tip would probably tell you that, but nonetheless, your DNA will too. And about taste and cilantro and lactose intolerance. Maybe those are just kind of for fun. Then there's this pharmacogenomics. There are 50 medications whose response in you is going to be influenced by what particular versions you have of the 7 genes that you see listed here, all of which have funny names. And then for 59 genes, where we know that people who have a misspelling in that gene are in fact at high risk of a significant disease for which there is an intervention available. You will hear about that too. And that's about 3% of the population gets that information back. Things like BRCA1, BRCA2 are on there and others as well. All of that comes with the effort, and that also comes with a genetic counselor to help you weed through it if you're one of the folks that gets that kind of an answer. So all that's happening, and again, I think the people that are part of this are having quite an interesting experience, and we are having an interesting experience learning from them about how they incorporate this information into their own healthcare. It's also a great resource for researchers because it's such a big study of these people. Already there are 245,000 that have had complete sequencing of the genomes. That's the largest collection of whole genome sequences in any study anywhere. And that makes it possible for people doing interesting work here to come up with something. Here's a cool example. There's a particular condition called primary hypothyroidism. These are people whose thyroid gland does not produce the thyroid hormone the way it should. We haven't really understood exactly what's going on. This is an autoimmune disease. Way back in 2011, a group of 5 sites working really hard took 2.5 years with 40 people to discover one place in the genome. Here's another Manhattan plot where there's just something on chromosome 9 that gets into the statistically significant zone. One locus. Now, just last year, this guy, Anav Babar, who's a post-bac student, just sort of taking a year between undergraduate and graduate school or medical school, decided he was going to get credentials to work on the All of Us database, which is not hard to get. You have to make these various promises about not figuring out who people are. And in 6 weeks, he found 10 of these contributions, so-called loci, in this diverse population represented by all of us across the genome. Now, notice the y-axis here goes from 0 to 80 instead of 0 to 10. So these are highly significant. There's that same region on chromosome 9 that the group knew about in 2011, now much more strongly, but there are all these other peaks too. Because now the size of the database is about 10 times bigger. And of course, the data is all accessible. It's just a small example of how power is so much enhanced by having scale. And a lot of what all of us is doing is providing scale that we never had before. So again, if you're interested, join allofus.org. You can be part of that adventure. So that's a lot about the basic science and the genomics of discovering how it is that these disorders are somehow encoded in various ways in the genome. But what about doing things therapeutically? Don't have a lot of time to go into that. That would be a whole nother talk, but I'll touch upon a couple of examples. And I think we have to start with cancer, because cancer is a disease of the genome. It happens because of misspellings in genes in the genome that have some role in telling cells whether they should grow or stop growing. Cancer happens when cells grow when they should have stopped. There are genes called oncogenes that are basically the accelerator in your cell's version of a car. And if that oncogene gets activated, it's like the accelerator is stuck and the cells just grow when they shouldn't. And then there are other genes in your cells that are like the brakes, and they stop the cells from growing. But if you inactivate them, then you crash into the barrier because your brakes didn't work or your cells grow when they shouldn't have. We are learning so much about the genes involved in cancer. If you are somebody who just had a diagnosis of cancer here at the top of this diagram, then getting a sample of that tumor, or even getting a blood sample that has some of the tumor DNA in there, is going to tell you something really important about what's driving that cancer in you. And you will be unique from everybody else because every cancer is a little different. So you'll be able to say which genes, which pathways have gone awry that are causing your cells to grow when they should not have. And that gives you a profile and interpretation. And these days, if you're in a cancer center that has a lot of experience, that's going to bring your case to a tumor board. It's going to look at all the data, look at all the possible drugs or immunotherapies that might be available to you, and try to match the specific characteristics of your cancer as determined by its DNA analysis with a range of therapeutics that might be available. And that is precision oncology. You do the targeted therapy and the trial matching. And that might be a chemotherapy, but not a carpet bombing version, but a very smart bombing version that goes specifically to the problems in your cancer that drug can attack, hopefully with a whole lot less toxicity. And sometimes that will also be a way to activate the immune system to wake up and see those cancer cells that they have somehow missed and activate them to go after those. Again, if any of you were to develop a cancer today, I would think this is the kind of workup you would want. It's still not happening in every place, but certainly in tertiary care centers, it is becoming the standard of care. Precision oncology, as opposed to the one-size-fits-all, is saving a lot of lives. It's one of the reasons the deaths from cancer are now dropping. Each year, we're down now 25% lower in deaths from cancer than we were 20 years ago. Immunotherapy is an important part of that. Again, this has been just a dramatic set of advances where patients with conditions like metastatic melanoma, like President Jimmy Carter, who we would have thought that was kind of the end of the line 7 years ago when he announced he had melanoma metastatic to his brain and his liver. And yet immunotherapy brought to bear on this was able to basically find the cancer and get rid of it. And while he's still struggling a little bit on hospice, it's not because of that cancer. Here's a dramatic example of a case treated at NIH where you can see, in this case, a case of individual with pancreatic cancer, one of the most feared versions of cancer that has spread to the liver. So on the left column here, pre-treatment, 3 different versions of a scan of the liver showing you all of these various spherical obstacles in the liver. Those are all metastases of pancreatic cancer, probably hundreds of them once you go to the smallest possible size. This is the picture of somebody who might have a couple of weeks to live, but instead was enrolled in an immunotherapy trial that basically found the rare immune cells in this patient's system that were already trying to attack this cancer, but they were outnumbered. Find those cells, expand them to the billions, reinfuse them into the patient, and 6 weeks later, look what happens. So that's the kind of thing that gets you excited, that maybe even stage 4, which is usually the kind of thing where you start getting your affairs together. There is no stage 5. Maybe not necessarily. Might that always have that kind of outcome? Look at this kind of thing that's now possible. Combinations of immunotherapy and genome-based drug therapy are also being tried. There's so much potential here. Some of it also depends upon using gene editing to basically take your immune cells and take them to school. Not just undergraduate, take them to graduate school. Teach them what they were missing, that there were these cancer cells out there that they were somehow not smart enough to recognize. Okay, get them educated to do that. And this brings me to CRISPR. CRISPR-Cas9 is like the buzz, and it should be, that all of us have been excited about now for going on 8 or 9 years since this emerged from studies of yogurt and bacterial viruses and became the subject of a Nobel Prize to Jennifer Doudna and Emmanuel Charpentier. Basically, this is an enzymatic system that is incredibly elegant because it can be basically programmed to go and find in the genome, and the genome's a big place, 3 billion letters, it can find this very precise target that it will then go and discover and do something to. And the initial version basically goes and makes a double-stranded DNA break. It just cuts the DNA, basically inactivating that target. It's getting much more sophisticated, though, and a colleague of mine, David Liu at the Broad, who's a remarkably creative inventor of enzymes based on really solid knowledge of chemistry, allows you to not just go and do a double-strand break, but find that one letter that you want to fix and do a find and replace, kind of like you would on your word processor in a very elegant way. And that is starting to work in a remarkable fashion. So that sort of brings me to, okay, what would you do this for? But first, I just got to show you this really cool image of what this looks like. In case everybody's nodding off, you'll enjoy seeing this as a little break between my static slides. This is a video from Feng Zhang, also at the Broad, who's one of the innovators in CRISPR-Cas. So what you're looking at before I started moving here, this is this Cas9 enzymatic molecule. And you can see sort of hiding inside it is something that's an RNA, a so-called guide RNA, that has its sequence that's designed to match in a Watson-Crick base pairing with its target. That's how it gets its specificity. But Cas9 has the ability to do the searching, and then once it's found its target based on that RNA, it can then do its thing. In this case, it's going to make a double-stranded cut. So watch this happen here. So here it is. It's coming in. It's searching. That guide RNA is looking to see, am I a match? Oh boy, it looks like I am. You can see that's all matching nicely in a base paired fashion. And now Cas9 sees that, and there's that first cut, and there's the second cut. And now you've got a double-stranded DNA break. And again, in this simplest version, what then happens is it's got to figure out how to repair itself somehow, so some random nucleotides get in there, and you end up with what is basically a stop in that gene. You've knocked out its function. And there are plenty of places where that's valuable, but there are lots of other times where what you want to do is to do a fix and not a break. And that's basically, again, what David Lewis and others are figuring out how to do. So this has revolutionized what we do in the laboratory, and all of you who are part of the affiliation of Christian biologists, I would bet a lot of you are using CRISPR-Cas in your own research. Certainly in my lab at NIH, we're doing this every day to try to understand how the genome works by being able to manipulate it. But we also want to apply this therapeutically. And here's where things get a little dicey, and people, I think, sometimes need a little bit more explanation about, wait a minute, what are you actually editing here? The promise of this for disease treatment is what I would call somatic cell gene editing. You're focusing on the organ system where the correction is needed. This is not affecting the germline. It won't be passed on to children. We're already doing this for cancer immunotherapy. And just in December, FDA approved this kind of somatic cell gene editing as a curative therapy for sickle cell disease, basically taking bone marrow cells out of the body using CRISPR-Cas to knock out a particular transcription factor, which turns out to be really important because without that, the individual's blood cells continue to make fetal hemoglobin, which is curative for sickle cell disease. There are a few experimental protocols now approaching this for in vivo gene editing, where you actually put the gene editor into the body. My own lab is doing that for a rare disease called progeria. And this holds enormous promise for the cure of many genetic diseases. But certainly, you can't look at what's happened with sickle cell disease without being incredibly gratified. I did not think in my lifetime we would see a genetic cure for sickle cell disease, and we have. This is Victoria Gray. I think she was the very first patient enrolling in the sickle cell therapeutic trial using CRISPR. With a great deal of uncertainty how that would work, I've gotten to know her pretty well now. This was 2019. This worked extremely well. This is Victoria actually walking the halls of Congress, being a very good advocate for how important it is that other people should have access to this as well. As far as we can tell, she's not benefited by this. She's cured. Basically, this has been a totally transformative experience for her and a few hundred other people who have now had this. And now that FDA has approved it, should be accessible to everybody in the US who has sickle cell disease, with admittedly a big challenge of the cost, which is $2 or $3 million per patient, and the challenge of doing this as an inpatient for about a month in a hospital because of the need to do a bone marrow ablation along the way. But of course, the dream here is, remember this diagram with just those 500 with therapy? These are all disorders where we know the misspelling. If we could figure out how to do this kind of in vivo gene editing, targeting the right tissue, the right gene with the right apparatus to fix the mutation, the find and replace model, all of these could be approachable. That scales really well if you can make it work. And there's a lot of effort now trying to do exactly that. But of course, I can't leave this issue, and I'm getting close to finishing the presentation here, without pointing out that while this is an enormous boon for a condition like sickle cell, where the treatment is focused on somatic cells, one does have to worry about proposals to actually apply this same kind of editing to an embryo, basically changing its DNA in a way that would affect not just that individual, but their future offspring as well. To be honest, it's hard to identify medical applications that are very compelling in this situation. And of course, it starts you on what would be classically called a slippery slope from treatment to enhancement. This is no longer abstract. A rogue scientist in China a few years ago actually did modify embryos in two little girls who were out there somewhere to modify their genomes, despite the strong international prohibitions against that. Again, I don't know the justification for this. I worry a lot about the idea of carrying this forward for enhancement. Who gets to decide what's an enhancement anyway? And what are the safety concerns and the theological concerns? If we are in the Imago Dei, are we in the right place here to start to modify that by tinkering with the very instruction book for humanity? So those are profound questions. Safety risks unknown, future generations would be affected. Will that separate between the haves and the have-nots? What does it mean to be human if that can be altered? Are we going to be fearfully and wonderfully remade? Are we taking on a role that justifiably belongs to the Creator and not to ourselves? I don't have easy answers to those, except to say that for me personally, I think to cross that line into germline gene editing is not something we should be doing now, and maybe shouldn't be doing ever. But there's a lot of different opinions about that. Finally, I want to say all of this great advances that are happening with medical research are extremely exhilarating. It's just like an amazing time for making discoveries and understanding how life works and how disease happens. But unless it actually gets to the patients, then we shouldn't be all that gratified about it. And so I want to quickly tell you about an example where that last mile, which should have been easier than it is, it turns out to be really hard. And it's sort of a wake-up call for me as somebody who's primarily been involved at NIH in medical research and hasn't thought quite as much about the implementation, that last mile. And this is a disease which was mentioned in the introduction that I am now obsessed about, I will confess. A disease called hepatitis C kills 15,000 Americans every year, and 2.5 million, roughly, of us are currently infected with this virus that attacks the liver. 40% of them don't even know they have it. The epidemiology would say a bunch of those are baby boomers. Most of those got infected with a blood transfusion before we knew how to screen for this virus. And then there's a bunch of millennials who got infected with a dirty needle, maybe a tattoo needle, maybe intravenous drug dabbling that they did. Once you've got this incredibly infectious virus, it is working away on your liver over the course of years to take you forward without much in the way of symptoms for the first decade or so into hepatitis, ultimately cirrhosis, and ultimately liver failure and death, or death from liver cancer. This is the most common cause of liver cancer. And it's all preventable because we have not just a pretty good treatment, we have a cure. This is like one of the most amazing achievements of medical research in the last 20 years. One pill a day, 12 weeks, 98% cure, almost no side effects. So why is this a problem? We've had this now for 7 or 8 years. Well, there are all these barriers that have kept a lot of people who need this from getting access. It's very expensive. It's still about $24,000 for that 12-week course, even though it's gotten a little better than the $90,000 where it started. It's still out of reach for a lot of people, especially if you don't have insurance. A lot of people don't know they have it because of the lack of symptoms. Insurance puts up barriers because they don't want to pay for the cost. Oh, yeah, you have to prove you're sober for a year. We need you to see a specialist. You need to prove you already have liver damage. That's so backwards. And the primary care physicians aren't necessarily comfortable treating this, although it's actually pretty straightforward. And a big issue is, where are a lot of the people who need this? They may be people who have injected drugs. They may be currently injecting drugs. They may be in the prison system because of the relationship to injectable drugs. They may be uninsured. They may be on the Native American reservation. They're marginalized people, but there are people too. They're our family, and we have a chance to cure them. And how can we walk away from that and say, well, that's just okay, especially because if we don't figure out to help them, we're not only dooming them to a really terrible future. And my brother-in-law died of this disease, so I watched what that was, and that was grim over the last 2 or 3 years of his life before we had these drugs. But we're also going to rack up a huge bill for our healthcare system, taking care of people who could have been cured, but instead now need a liver transplant or treatment for liver cancer. So there's a plan. And when I was serving as the president's acting science advisor, this emerged. And I'm still asked by the president to be the lead for the White House on getting this actually implemented. It has 3 parts. One part is a basic practical one, is that the testing process is very clunky right now, and it takes you 3 visits to the clinic before you know you need the treatment. That's crazy. Let's have a point-of-care test that gives you an answer in less than an hour, and then once it's positive, here's your bottle of pills. 1 visit, and you're testing and treating. And that is actually something we're working on and should be available as soon as July of this year. And then let's make the drug free instead of so expensive. And this is what's called the Netflix model, where the companies agree to take a particular lump sum, in return for which they make the drug available for free to anybody on Medicaid, anybody who's uninsured, anybody in the prison system, and anybody on the Native American reservation. This worked in Louisiana. It seems like it should work for the whole nation. And then we got to provide some resources to make it possible to deliver the testing and the treatment for marginalized groups. This is all very well worked out. This is one slide that is actually undergirded by a very detailed 5-year plan that I'm quite convinced would be successful. So why haven't we done it? Well, we are close to getting this to the point where it could actually happen. Legislation is finalized. There's strong Senate support, bipartisan. Senator Cassidy, Republican from Louisiana, Senator Van Hollen, a Democrat from Maryland, strong support from advocates. What we're really waiting on right now is for the Congressional Budget Office to decide whether this is going to save money. We've had a Harvard economist look at that who says this will save $8 billion to the federal government in 10 years and more after that. But until CBO says that, it's still up in the air. And then if they say, yeah, it's going to save money, I think we could probably get a vote of both houses and the president is ready to sign. And the potential result, tens of thousands of lives saved, tens of billions of healthcare dollars saved. This feels to me, for a Christian's perspective, like something we are called to. You know, this verse from Matthew 25, this is the words of Jesus where he's talking to the disciples. Then the king will say to those on his right, this is about the judgment day, come you who are blessed by my Father, take your inheritance, the kingdom prepared for you since the creation of the world. For I was hungry, you gave me something to eat. I was thirsty, you gave me something to drink. I was a stranger, you invited me in. I needed clothes and you clothed me. I was sick and you looked after me. I was in prison and you came to visit me. And the righteous are wondering, what was that all about? When did we see a stranger and invite you in? When did we see you in sick or in prison? The king will reply, truly, I tell you, whatever you did for one of the least of these brothers and sisters of mine, you did for me. I can't help but reflect on that verse when I see what we should be doing for these brothers and sisters with this disease that we could cure. And how can we look away and say it's just not convenient? So, conclusion, I do want to point you to BioLogos as another place that has a lot of conversations that you all, I think, also are deeply engaged in, and many of you know this site, bringing together science and faith, and BioLogos now especially focusing their attention also on climate change. And I understand you're having Catherine Hayhoe come and speak to you soon, who is just fantastic. And all of this, and Deborah Harzma, with the president as an astrophysicist, brings a particularly wonderful perspective to BioLogos as well. And a particular verse that I'll finish with, because I find it so encouraging, and particularly in this point in our own country's history, where everybody seems to be kind of mixed up about how did you decide who to trust and what is true. Jesus is telling us, love the Lord your God with all your heart and with all your soul and with all your mind. I think that's what the affiliation for Christian biologists is all about. So I'm glad I've had the chance to speak to you, and I fear that I have run 13 minutes over. If we need to run a little bit longer on the Q&A, that would be okay, Brian. But with that, I will say thank you all so much and look forward to the questions.
Francis, thank you for that informative and inspiring presentation. So one of the take-home lessons from the Human Genome Project and its various spin-off projects is that race is a social construct rather than a biological category. Yet there clearly has been a lot of emphasis on diversity in this All of Us research program project, where, as you noted, nearly half of the individuals enrolled in the program are from underrepresented racial and ethnic minorities. So can you unpack for us why is there such an interest? You know, if race is really a social construct and not biological, why the focus on diversity then?
Yeah.
And how does race factor into precision medicine?
It's a great question, Brian. I think all of us actually got in a little bit of trouble with the publication they put out in Nature about what they'd learned about the genomes of those they'd participated. There was a diagram in there using something that was aiming to identify similarities and differences between participants. And it was possible to be able to put sort of clusters out there based upon genetic information that reflects ancestry. That's probably not surprising. 23andMe and Ancestry.com would not be able to tell people roughly where their ancestors came from if there were not reflections in the genome about the frequency of particular variants. That is an indication of what that particular individual has inherited down through the generations from their particular part of the world. But let's be clear, we are all one family. That's one of the gloriously wonderful outcomes of the Genome Project. anybody who tried to say there were multiple different independent origins of humanity that involve Homo sapiens, it doesn't work. We are all very much descended from a common set of probably 10 or 20,000 ancestors, probably in a certain part of East Africa, probably in the neighborhood of 150 to 200,000 years ago. We are all part of that family. But as we migrated around and variations happen in the genome, some of them random, some of them even selected for, like skin color or lactose intolerance, those variations basically became part of our ancestry. And while we are incredibly similar, with 3 billion base pairs to look at, there's enough of those differences to be able to infer somebody's geographic ancestry with a fairly good amount of accuracy. But when we talk about race, what people are referring to is often much, much more than simply that geographic ancestry. It has a huge amount to do, much more to do with culture, with history, with societal practices. It continues to be a blurry area. I would point everybody to a report by the National Academy of Sciences just in the last year that tried to unpack this, and particularly for researchers, tried to make recommendations about how best to deal with these terms like race and ethnicity and ancestry that are fraught with a lot of baggage. I don't think it's correct to say that what one uses as terms as self-identified race is completely disconnected from genetics. There's a connection, but there's a whole lot more there that's probably more important for the individual's health and self-identity than just their DNA sequence.
Francis Collins, thank you very much for this great talk. I must admit, I voted for you for president last time around. You seem to be the most moderate candidate, the least polarizing.
I didn't know I was a candidate. Wow.
Well, anyway, I've been intrigued by this research on Neanderthals and what genes came down through all the years of evolution and which did not to us. And it's the favorable ones that we still have and the unfavorable ones we lost for the situation we were in. So here's a hypothesis. Can we learn something about where this COVID came from by taking your chromosome 9 data? And here's a hypothesis. So the kinds of bats that had the COVID-like virus genes weren't around where Neanderthals were, so they didn't have to develop a resistance to it.
That's an interesting hypothesis. I think probably Neanderthals having encountered bats would have happened, but maybe not in Southeast Asia, because it does look as if the interactions between Neanderthals and Homo sapiens were largely in Europe, and you don't see as much Neanderthal DNA in other parts of the world. I would bet, though, that whatever that segment of chromosome 9 is that seems to play a role in severe COVID illness is probably not specific just for that one coronavirus, but it is probably a more general reflection of how the immune system responds to an invader. We don't know enough about that yet, but people are certainly looking. And I think you're right, though, that it is teaching us something historically about what these various populations were exposed to and the delicate balance they had to have between responsive to an infection, but not too responsive, where the immune system became more of a problem than the solution. And maybe that says something important about infectious disease history over many, many, many millennia, but it's pretty hard to sort out at this point. Maybe we'll get a better handle on it over a while. I do think, though, that it's pretty intriguing that if you'd asked most people 20 or 30 years ago, is it likely that we'll ever know whether Neanderthals and Homo sapiens had interbred, people would say no. And now everybody's absolutely convinced that, yes, that happened, and there's the data.
I just wanted to first thank you for that really beautiful presentation. I mean, you gave us so much to think about. It's almost hard to keep it all in mind, but That was just wonderful. I have a fairly straightforward question about something you said early, which was related to the 600 genes in type 2 diabetes. Roughly what percentage of those were in coding genes and what were in regulatory regions? And can you give us any insight into that?
Yeah, I can, because we've looked at them very carefully. 95% of them are in regulatory regions, 5% are coding. So that tells you a lot about what's really going on. And it's hard, Cy, as you know this, because you can say it's not in a coding region, but then you have to figure out, well, how does it work? It must be some regulatory signal. It isn't always the case that it's regulating the closest gene. Sometimes because the genome is not arranged in a nice linear fashion, it's all three-dimensional. You could have a variant that's actually having its main effect on something that's a million base pairs away, but just happens to be folded up next to it in the interphase nucleus so that these things can interact.
Hi, Francis.
Hey, Daryl.
I was wondering about the all that non-coding genome. So the exome has almost become old news now. And I was wondering if there are any therapies that might come along from regulatory genes. I mean, we know the RNA vaccine story. Are there other RNA cures that might be coming along?
It's a great question. This is still kind of the dark matter of the genome, therapeutically anyway. I know people who are trying to figure out how that works. We have a large and growing collection of information about enhancers that play a significant role in activating genes nearby, often in a tissue-specific way. And some of those are involved in cancer, like a famous enhancer that's a long way away from the MYC oncogene, but plays a big role in whether or not it's activated in cancer. But it's been relatively few examples of people trying to utilize that therapeutically to try to block that effect. You could try that with an RNA therapy. You'd have to count on that regulatory signal being sufficiently accessible that the RNA could find its way to get in the middle of it and block whatever it's doing. And that's a bit of a steric problem, I'm afraid, for a lot of these. So much of that still seems a little out of reach, but I think you're right. There's a great potential there if we had the right chemistry to be able to open up the window of targets beyond the traditional ones of coding genes.
And if I could ask a second question, or maybe ask for a little bit of history, one of the things that I really admired was the way the Genome Project started up with allocations for ethical studies. Could you tell us a little bit about what came out of the ethical study budget from the Genome Project? I
was a big fan of that. This was the ELSI project that was called Ethical, Legal, and Social Implications, ELSI. And it was 3 to 5% of the overall budget, pretty much 5% for most of it, which doesn't sound like a lot. But when you consider this was a pretty significant budget for the Genome Project, it became one of the largest investments in ethics research that NIH had ever mounted, and continues to this day to be a significant contributor. I could point to a bunch of things. I guess the one that maybe had the most significant impact, because it made it all the way to legislation, was this conclusion that genetic discrimination really ought not to be allowed. You don't get to pick your DNA, so it shouldn't be used against you. And that led to a lot of scholarship, some policy papers, ultimately over 12 years working with the Congress, the passage of GINA, the Genetic Information Non-Discrimination Act, signed by President George W. Bush in 2008. And I was proud to be in the Oval Office when that signature happened, and I have a copy of it on my wall here with his name scribbled at the bottom.
Hello, I'm not Christopher Plotchik. I'm Dr. Ann Patterson. I'm here with a small watch party. We're in the dark with some students and faculty enjoying the presentation. I want to thank Dr. Collins profusely for the enjoyable presentation. I've been following the Genome Project since it started, and thank you very much. I could ask a long list of questions, but I'll keep this very short and non-technical. Just, we're all located in a very rural area, and I couldn't help noticing in the presentation on all of us that there's very heavy representation in California and the Northeast. And I know you were talking about cultural factors, having a Fitbit-like thing. So there's not just genetics, it's looking at other things. And I wonder if that's a concern or something that anyone's trying to address.
It absolutely is. I was trying to quickly pull up the All of Us diagram there to remind myself what the representation is in rural communities. It's only 7.7% of the participants are rural area residents, and I know they aim to try to get that a little higher. And certainly you're right that some of the things that you would like maybe everybody to be collecting is not going to be so easy. I will say All of Us makes Fitbits available. They send them to people who want them, so you don't have to buy your own in order to try to make this diversity more well represented with some of the data that's there. They have a very active patient participant group who have been very engaged in this kind of outreach. They run mobile units that go out into rural communities who would otherwise, I think, not be likely to be near a place that was doing enrollment, and continue to come up with creative ideas like that. COVID did not help, I must say. The enrollment during 2020 through 2022 was slowed down a bit, but it's really picked up now. And certainly I would hope that that would also be reflected in a big push in rural communities. We really do want to know what's happening to all Americans and not just people in the big cities.
And what would you say is the most important insight that scientists have gained by having to deal with the science denial that emerged as a result of the pandemic?
Oh, Terry, that's a really sobering and important question, isn't it? I think we've all seen what has happened in the course of the last 4 years or so, with multiple surveys documenting the distrust about science that has grown in the public. Ironically, just at the moment where I think objective views would say science rose to this challenge in a pretty remarkable way, with the successful development and testing of vaccines that turned out to be highly effective and safe in just 11 months. But all of the other information that people got and their gradual becoming disillusioned with the public health system has led us to be in a very bad place. It's hard for scientists, I think, to feel necessarily that they have a path forward to try to convince people of what is true when so many people are now residing in particular information bubbles, and it's difficult to break through those bubbles if you're providing information that doesn't seem compatible with what they wanted to hear. And scientists are all too often being also seen as elitists that are sort of out of touch. Insights, I do think, Terry, that, and I'm speaking for myself here because I was one of those called to try to speak frequently to the public about what we knew about SARS-CoV-2. I think we could have done a better job when we were doing that to make it clear that there was a lot of unknown information, that we were doing the best we could with the data we had on a given day, but that it might need to change. This could have been a great lesson about how science works, that you have incomplete information, you try to get something better, you draw a tentative conclusion, you have a hypothesis. But in the course of a crisis where people are dying all over the place and you have cooling trucks pulled up outside of the morgues to handle the dead bodies, you have this sense of urgency of trying to convey to the public as best you could what you think might save lives. And sometimes it turned out wasn't necessarily right. And then the public begins to think, these guys don't know what they're talking about. Mm-hmm. Or maybe they're even jerking us around. And along the way, with all the misinformation that was coming at them from multiple directions, some of them political, I'm sorry to say, as well as people who were genuinely trying to make money off of this by spreading information that was demonstrably false, people had a very hard time sorting this out. And science communication was kind of back on its heels a little more than it should have been, trying to play catch up with all of the other claims that were out there, many of which were not based upon evidence. It's very sobering to see how this has happened. The future of our nation really depends on our recovering some sense about the difference between opinions and facts, and the ability to be able to understand how science can, in fact, make mistakes, but is self-correcting. And if you're trying to understand something about a virus that's killing people, science is your best hope of getting an answer that's going to get you where you need to go. But again, we got a long way to go to try to recover that. I've written a book about truth and science and faith and trust, which will be out in September, where there are a fair amount of hand-wringing on my part about this, but also some suggestions about actions that maybe we as a society could take to try to turn this around, because this is a very bad place for the most technologically advanced country in the world to be, where people can't even quite agree about what is a fact anymore.
Okay.
My name's Catherine. I'm a student. I was part of a watch party at Lipscomb University.
Okay.
And I thought everything that you said was super interesting. I'm not a science major. I just thought it was really fascinating. But looking more at the healthcare side of things, you mentioned a lot of the costs throughout the presentation. So relating to the financial side of what you were talking about, how feasible do you think this concept of personalized medicine is? Like, how soon do you think these things will be implemented into our healthcare system that already has so much cost built into it?
Yeah, it's a great question. And you're right. Our system has a huge amount of cost built into it, for which we are not necessarily getting great value. If you look at a scatter plot, just think of a two-dimensional plot of life expectancy versus dollars per person that we are spending on healthcare. You might expect there would be some relationship. There is across the world some relationship. The countries that have spent more seem to have better longevity. But the big exception, the big screaming outlier, is the United States of America. Our longevity is not nearly as good as in Scandinavia or in some parts of Asia, and yet our expenditure per person is off the charts. So we are doing a lot of wasteful medical care. We are investing very poorly in prevention. Other countries do a better job of that. Our system, though, is wired to think about short-termism. And every time you come up with a great idea about prevention, which is going to save money 10 or 15 years from now, but cost money in the next few months, it gets shot down. The insurance companies say, I don't want to pay for that and have this person who I'm taking care of on my insurance policy move to another insurance policy later, and then I won't even get the benefit because they move on. And the government, unfortunately, also thinks things in short term. I talked about this with hepatitis C. Unless the Congressional Budget Office says that an intervention is going to save money in 10 years, why is it 10 years? No good reason. Then it is really hard to get anything done. So it's our system that is getting in the way. I think with precision medicine, the task will have to be to make the interventions really cheap so that it isn't a huge burden in the short term. And that's a place where it's starting to get pretty reasonable. After all, the first human genome cost us $400 million. You can get your genome sequenced now for under $500. This is a whole lot less than an MRI scan. The idea that we don't currently have genome sequences for everybody in their medical record doesn't make a lot of sense. As soon as we can convince the insurers and Medicare and Medicaid that this is a good investment, then that will start to happen. Then once you've done it, I mean, think about this. Once the information is there in the record, to insert that into your prevention program or your pharmacogenomics program doesn't cost anything except a little provider time. You don't have to do another test. It's been done. So I'm encouraged by the scenario, but worried about the incredible slowness of our system to actually embrace things like this. But it's a great question.
Thank you, Francis, for your great presentation. I think you can see it, I am an All of Us member, and one of the earlier ones, and I've got all my traits that have been identified, and I'm happy to say that I don't have any of the disease variants that are been tested. But I'm wondering, are there any more traits? Are there any more disease variants being planned to release to the public, to the participants?
Yeah, there are, Carlos. There's a big discussion going on about what would be the threshold for telling people about things that they would want to know about. The 59 that we started with was a very carefully curated group developed by the American College of Medical Genetics. But they've already revisited that, and it's now up to at least, I think, 80. And so there will be a refreshing here as we get more data and we find more things. Not that I think they're not basically offering to tell people information that you can't do anything about, but the ones that you can, it feels like an obligation that if people have trusted us to be part of this, we have to tell them what we've learned. So it will be growing. You expect follow-ups.
Yeah, just for a minute, I wanted to remind everyone that this summer, the ASA annual meeting will be in Washington, and we're delighted that Dr. Collins has agreed to give an important plenary lecture at that meeting. So if this is any indication of what that will be like, I think everyone will want to come. I would also say there was a question about race and genetics, something which I actually had done research on, and we're very fortunate to have Dr. Charmaine Greuel, who is an expert in that field. She is going to also be a plenary speaker at the conference. So I'm looking forward to that, and I think we all should too. And I, along with Mike Beidler, are co-chairs of that meeting, along with the home office of the ASA. And so if you have any questions or issues that you want to bring up, feel free to send me a note, or Mike, or any of the people in the home office of the ASA. So I hope to see you all then.
Dana, you mentioned something in the chat. You were wondering about are there so we know there are 2 watch parties at least, one at Lipscomb that has 45 students and 3 faculty, and then one at Williams Baptist University that has 9. Is that, you just wanted me to mention that? Is that okay?
Yeah,
That's great.
Yeah, we're glad to have watch parties. That's wonderful. Okay, well, I just want to thank everyone for joining us for this presentation of the Affiliation of Christian Biologists. And if you're interested in learning more about the Affiliation of Christian Biologists or its parent organization, the American Scientific Affiliation, I'd invite you to send an email request to asa@asa3.org. Again, that email address is asa@asa3, that's the number 3.org. And now I would like to invite Carl Pinkham to close us in prayer.
Let's pray. Lord God Almighty, creator and sustainer of the universe, thank you for the joy of being alive in these exciting times. Times when our knowledge of your world is both underscoring the truth of your word and placing us on the brink of an ability to reveal your immense love for us as individuals through individualized medicine. Thank you for the example of leaders like Francis Collins and his clear dependence on you in all he does. Lord, no matter what we are called to do, may we all similarly glorify your name and the knowledge we convey to a questioning, wondering, and seeking world. Pray this in the name of the Father, the Son, and the Holy Spirit, the God who is 3 in 1. Amen.
Amen.
I should have mentioned also that Carl is the vice president for the Affiliation of Christian Biologists, and I really feel like we make a great team. So thank you, Carl. And Francis, thank you for a wonderful presentation. thank you all for attending tonight, and wish you all a great evening.